Hello everyone, my name is Renama, and today I'm presenting at the third RNA virus discovery symposium. I'll be talking about a newly detected narna-like virus that I named Glossinomyces trisopsiformis. The name Glossinomyces comes from Glossina, the closest fly, with myces, a fungus-like ecological context within the tsetse holobiont, and the species name, the trisopsiformis, means trisopse-like, which refers to the closest database relative, the Trisops narnavirus. And today, I will walk you through this work in 6 parts, which is background, hypothesis, methods, results, discussion, and the conclusion. So I will first start with the ecological context. The virus was discovered in the RNA sequencing datasets derived from tsetse fly, the Glossina mositans. These flies are medically important because they transmit the Trypanosoma brucei, which is the causative agent of the African sleeping sickness. Because of this, the Glossina has been extensively studied as a whole system, including itself, the parasites, the bacterial endosymbiont, and the polycharacterized, the microbe, eukaryotes. This makes it a particularly rich system for discovering the hidden viruses. RNA viruses are typically characterized by compact genomes and rapid evolution. Among them, the Narnaviruses represent an extreme case of simplicity. They are traditionally described as positive sense, single-stranded RNA viruses, around 2 to 3 kilobases long and encoding only one single open-reading frame for an RNA-dependent RNA polymerase, and lacking both a capsid and extracellular variants. They infect the unicellular eukaryotes, most commonly fungi. However, this textbook view has begun to crack. Recent metagenomic studies have reported long complementary-strand open-reading frames in several narnaviruses. These viruses may encode more than just one RNA-dependent RNA polymerase, although the biological function of this additional ORF is unclear. Based on this background, I ask 2 main questions. First, what is the genomic composition of this newly discovered virus? And second, does it confirm to be the classical single ORF narnavirus, or does it encode an additional functional capacity? My hypothesis was that the virus belongs to the narnaviriid, but deviates from the classical single open reading frame architecture. First, to determine the taxonomic placement, I used BLAST-P and the Serratus Plum ID analysis. All close matches belong to the family Narnaviriid. Second, to construct the virus genome, I combined the novel assembly with Cap3 of the long RdRp containing conjugates, and also I used the reference-guided assembly to combine those short conjugates with the most closely related nanoviruses. The open reading frames were predicted using the NCBI ORF finder, and functional annotation was performed with BLAST-P and Interproscan. So now I will move on to the result. First, the ecological signals is highly constrained. Across all datasets, the virus appears in 41 SRA runs, spanning 6 bioprojects and 3 countries. And crucially, it is found exclusively in samples derived from Glossina mositans. Within the fly, it appears across a wide range of tissues, including the gut, the bacterium, the parasite-associated samples, and even the larva milk. So this distribution argues strongly against the infection of a specific insect tissue or the parasite. Instead, I think the most logical explanation is that the virus infects a unicellular eukaryotic microbe, which is broadly distributed across the tsetse holobiont. So the second key result is about the genome assembly. The assembled genome is 2,342 nucleotides long and encodes 2 major open reading frames, 760 amino acid RdRp on the positive strand, and also 744 amino acid open reading frame on the complementary strand. The RdRp contains all the conserved motifs A, B, and C, and AlphaFold 3 predicts a classic polymerase field with an interact catalytic groove. And the reverse open reading frame shows no known domains. However, its uninterrupted length strongly suggests that there is selection against the stop codons. Similar reverse open reading frames have been reported in other insect-associated narnaviruses, supporting that this idea is a recurring genomic strategy. Together, these results challenge two long-standing assumptions. So first, the narnaviruses might not be truly single or ORF viruses. Instead, many of them appear to encode a hidden protein on the complementary strand. And one possibility is that the translation of this reverse ORF influences the stability or the accessibility of the native strand RNA during the replication, maybe modulated by the RNA duplex formation or interacting with the host factors. And this supports the recent proposals to subdivide the narnaviruses based on the complementary strand coding, placing our new virus within the alpha narnavirus group. And second, the virus ecology cannot be inferred solely from the sequenced host. Although detected in the insect dataset, our new virus, I think, is more consistent with infecting a microbial symbiont, not the insect itself. So, to conclude, our Glossina trisopsiformis is a newly detected narna-like virus widely distributed across the Glossina morstens datasets, and its genome encodes both a classical RdRp and a long reverse strand ORF, indicating that there's dual coding capacity. And the virus is most likely to infect a unicellular eukaryotes symbiont within the testis fly, rather than the insect or the parasite. And these findings suggest that the hidden coding strategies may be common in the ultra-minimal RNA viruses. So, taken together, this work shows that genomic minimalism does not necessarily imply biological simplicity. And thank you very much, and I'm happy to take any questions.